Okay, so continuing with our AI theme and sort of tying in with some of the themes that our keynote speaker talked about Monday. Our next speaker is from the UW Great Lakes Bioenergy Research Center, GLBRC, Samir DaCosta, and he'll be continuing our AI theme. Thank you.
Maybe I should be in front. Can you guys hear me? Hey, I'm Samir. I'm a scientist in the Computational Biology Group at the GLBRC. Most of you don't know what that is, but I'll explain. I just want to say thanks for inviting me to give this talk. It's been great. I've listened to a few of the talks, and they've all been great. Unfortunately, when I was listening to the keynote, I was like, oh no, this is exactly my presentation, but with AI. And so then, luckily, I hadn't made the slides yet because why do something unless it's the last minute? So, so I made some changes, but it is what it is. Just let me talk a little bit about GLBRC. It's the Great Lakes Bioenergy Research Center. And technically, I've been part of GLBRC for several years, but I only officially joined in December. So the mission is basically creating biofuels and bioproducts. And the key idea is that we want to create biofuels from non-food plant material. So that's very important because we don't want our fuels competing with our food. And we want to do that in a sustainable, like an economically viable way so that we can kind of make industrial processes and use all this plant matter that's not being used and just wasted and use it to create biofuels. And right now, this is all being done using yeast. But there is a promising microbe called Zymomonas mobilis. It's like a little alpha proteobacteria. And it's really good at turning sugar into alcohol, but it has some drawbacks, which is why it's not being used in industrial processes. And some of them are like, it works on simple sugars, but not on the more complex things. And it's not very tolerant of like certain kinds of solvents in the environment that are used in these industrial processes. So it needs a lot of engineering to get it to a stage where it can be used for these industrial processes. And that's genetic engineering. We like add genes, subtract genes, like tinker with it in any way we can. That's like we spend a lot of time just like playing with this. The problem is that we don't really understand, like we've sequenced the entire genome, so we know like roughly what the proteins are. We know there's about 2,000 proteins, so it's a lot simpler than E. coli, but it's very understudied. We don't know what half these genes do. We don't know how they function. We don't know like what happens when we take things out and put things in and blah, blah, blah. We want to kind of like, if you're engineering things for industrial purposes, you kind of want to like delete large circuits that are not used. Like, so for example, if this circuit is used for aerobic respiration and we don't need it, then we just like delete that circuit. And so once we understand more about what these genes do, then we are able to engineer this more effectively. This is a graph that I don't fully understand, but apparently everybody who studied biology will know in high school or something, or I don't know. I never saw this until like recently, but this is basically how everyone breeds. And this is the graph for Homo sapiens. And what I wanted to point out is basically the hydrogens these are protons that are pumped across this membrane. And these are complexes of proteins. So there's one here, it's a complicated complex, and here's another one with a lot of like subunits here, and a lot of one here, and they're called like complex 1, 2, 3. And then I want to go to the next slide, which is the same picture that we know of in our bacteria. So it's a lot simpler. It's a lot simpler. Some of these complexes like have analogs in the human thing, so we understand roughly what they're doing. But a lot of them we don't really understand. So over here, there's this guy I'm going to be focusing called cytochrome C, which is a small protein. It's got about only 100 amino acids. And basically, its job is to take electrons from one complex to another. And that shuttling of the electrons allows the complex to do its work and also allows it to pump protons across this membrane. And in Zymomonas, we just don't know what cytochrome C is doing after it gets its electrons from this complex. So this complex is similar to the complex that is this here. You can see cytochrome B here and cytochrome C1 here. And so that's this complex over here. It's got 3 proteins in it, and then they appear in 2 copies. So there's 6 in total. And we know that cytochrome C docks quite nicely into it, and then we just don't know what happens afterwards. And so some graduate student came and was, so as part of my job, I'm supposed to help labs with computational projects. And they came and they were like, well, what does this do? And I was like, I have no idea. And he was like, I have no idea either. And so we were trying to figure out how to get around, how to solve this problem. And just a spoiler alert, we don't have any answers, but we're just doing something anyway. So let me just say why this is a really hard problem computationally. Do I have the time? Any of that? Oh, you have time. Okay, thank you. So this is a really hard problem to solve computationally. So even though Zymomonas mobilis is only 2,000 genes, at half the number of genes as E. coli, we don't know what they all do. It's super understudied. But we basically don't know what proteins form complexes with other proteins. Like which proteins are forming a complex. We don't know that basic question, right? And so that it's become hard because you can't really answer the questions. You don't know what the complexes are. We have a few things in our favor. One is that I haven't put it on a slide, but it's a much simpler process. Bacteria are a lot simpler than like eukaryotes, so like humans and yeast and things like that. So it should be easier to compute what we're doing. The second thing is we can kind of predict where the genes are. So if you look over here, these genes are in these membranes and in this periplasm, but we don't want genes that are always in the cytoplasm because we know that most of what is happening over here. So there are programs that can help us predict which genes are in this region that are interesting. So we reduced the problem down to 580 genes. And now we're like, how do these genes like form? Which of these genes form a complex with other genes? But what we do have also in our favor, starting from 2019 onwards, is AlphaFold. And now this is the 3rd iteration of AlphaFold. So it's gotten a lot better. And AlphaFold basically is able to predict the protein structures directly from the sequence, right? So it's not 100% accurate, but it's like good enough for government work. And we're funded by the government, so it's fun. So we basically take their scripts and run that, and that can basically try and fold proteins into complexes. But just like to start with, if you have 2,000 proteins, the genes encoding proteins, you have 2,000 of them, you've got roughly like 2 million interactions. And we don't have enough GPU to like fold, to like try to like see if 2 million proteins dock, right? So we have to do other things. So that's why it's like this huge hard complex. And then after we figure out like, can these 2 things like dock, we also have the additional problem that AlphaFold is not always correct. So there's a lot more going into this. But the nice thing about AlphaFold is that the scripts are the pipelines are kind of split up into 2 things. And one is a data pipeline that we can do exclusively on the CPU. And then inference, which has to be done on the GPU, is just too big to be done without the GPU. So the data. So what is the data preparation? So this is all considered to be inference. So basically, the model is already built, and so I just need to like infer it, and that should just be like running a neural network. There's no reason for me to like do any data preparation. But the problem is that database of sequence structures, of protein structures, is only about 100,000 sequences. So when they built this model, they were not able to build this model entirely on protein structures. They had to search like really large databases to get sequence alignments, and that takes a lot of time. So this data prep step can be about equal to the inference step, even though if you're using a really fast and advanced GPU. So that is something to keep in mind. So what we did is we were trying to run a little test. So we're like, okay, let's just predict all 2,000 proteins of this of Zymomonas, and that took a week using infrastructure at CHTC, and we have like an in-house cluster that also uses HT Condor at GLBRC, and that's got 5 GPUs. I won't say what GPUs because it's a little embarrassing. But CHTC has got some big GPUs. So what we were doing was we would run the inference pipeline. We would select GPU memory based on the size of the problem, and then shuttle them to the right location based on because we had a lot of machines with low GPU memory and very few machines with high GPU memory. So we would basically decide where to run what based on GPU memory.
It's the 1.5 day on. So you use several GPUs, or can you do inference concurrently?
You can use several GPUs. We just use one GPU per protein, but for each protein we use one GPU.
I see.
But you can use several GPUs for one protein as well, and that scaling, it does scale up. So you do get some benefits, but it's not a linear benefit. Like running 3 GPUs will not get you 3 times as fast. It'll maybe get you 1.5 times as fast.
So you are running a single inference on multiple GPUs, or can you run several inferences on different GPU? It seems to me that you spend 1.5 days inference, right?
Yes.
So if you had 100 GPU, can you say inference, and collect the result?
Yes, these are all done in parallel. So we are doing them all in parallel. It's 1.5 days because we only had, because of my priority.
Yeah, okay. So if you had more GPUs, you would have, you could have pushed. I assume if you had more CPUs, you can push down the 5 days.
Yes, if I have, yes, I can.
Okay.
Yeah. If I have more, I can do more. Yeah, and that's one of the things I wanted to point out later as well. So the data pipeline took a long time because you have to search these 650 gigabyte databases. So we don't search them one by one. We search them in batches. So when we transfer them to a local machine, we basically do a bunch of them. Otherwise, we're looking at like 1 terabyte of, wait, tera? No, not tera. What is the next one? Peta. 1 petabyte. We're looking at 1 petabyte to do all these 2,000. So we have, that's a huge bottleneck. But the nice thing is that once I do this once, I can kind of skip the data pipeline in the future. So I wasn't too worried about this. I was more worried about this apptainer container. And so what happens is that when you build a container, the containers are built kind of sloppily, and they include all the programs that are there to actually build the program. And when you're just running things at inference, you don't need all that. So you can kind of skip a lot. So there's these 2-stage builds where you can kind of like make these containers a lot smaller. And now we can run 20, 30,000 runs that we're likely to do and not transfer as much data as we thought we were going to. So I want to come back to this. So what we did was we looked at cytochrome C, and we tried to see can AlphaFold put it in dock cytochrome C into these things, and also show that it will not dock into something like this or something like this. And roughly you're able to get that result. You can kind of like do cytochrome C against this one of these proteins, and cytochrome C against another of these proteins, and like try to see like what the docking scores are. But the problem is that unless you got the right proteins as part of the complex, and unless you got the right stoichiometry, which is like the number of copies of each protein in the complex, you are not able to get a very high docking score. So everything comes down to like, can we predict these complexes first? So here's what we did. We had no idea what the complexes are. It was too hard. We decided, okay, we can get complexes if we look at protein-protein networks and then like isolate these subnetworks. And then we say, okay, these subnetworks likely form a complex. And we can kind of brute force the problem a little bit. So we can get some complexes. But we expect, like, if you look at all the 2 million proteins, you've got like 2 million, so all the 2,000 proteins, you've got like 2 million interactions. And only 1 in 1,000 interactions roughly are likely to be a true interaction. So you're kind of looking for a needle in a haystack. You're looking for like 2,000 interactions out of 2 million. And it'll be super hard to find all of them. And so it will be a Hail Mary pass, and that's Eli Manning. That's what I got from Wikipedia for Hail Mary pass. And so we'll have to see if cytochrome C will form a complex with one of those complexes. Yeah. And if we're not able to do it, then we're just going to run more experiments and figure out experimentally how, like use our computational results to guide some experiments. It may not be solvable by computational results alone. And the nice thing is that whatever complexes we get, we can save for future work at GLBRC because we study this organism a lot, right? So it's not going to be wasted effort. So this is our computational plan. So this is basically roughly similar to the keynote, but like with using machine learning and AI to do protein-protein interactions. And I'm basically following these two things by David Baker. This is basically computing core structures of eukaryotic proteins. And so you can see nice, pretty pictures here. We don't know how many of these are true or not, but we expect some are true. And the thing is that our problem is slightly better because this was done for E. coli, this was done for eukaryotes. But bacteria have more genomic data available than eukaryotes, and the complexes are simpler. And also the interacting proteins are closer in the genome, so it should be easy for us to like go through this pathway. And we've started doing this. This is our workflow. That's the exact number of proteins we have. So we got to look at all bacteria and like kind of have a mapping for each protein to each other protein in every bacterial species. And sometimes it's missing, sometimes it's there, but when it's there, we got to find like a close match, a good match. Then we got to pair these protein alignments. And then once we've done this basically, and so we're in this step right now, and this allows us to skip the entire AlphaFold data generation pipeline because we can kind of directly specify with better accuracy than AlphaFold can specify what these alignments should be. And then we run coevolution, and in this case, we run an older machine learning technique called direct coupling analysis because we've got these 1.7 million pairs that we need to reduce down. We can't run AlphaFold on all of them. So we kind of run this, and this is pretty quick. This is 1 to 2 CPU hours each. So we're still looking at 1.7 million CPU hours. But on the GPU, it's a lot faster. It's basically 1 to 2. So we can run these in batches. I'm pretty sure this will go pretty fast. And then that just depends on our computing budget. Like if we get 20,000 pairs, we decide to like dock them with AlphaFold. And running this now is going to depend on the size of the pairs that we have. So the bigger, and the scaling does not scale up linearly. It scales up quadratically. So if you have 2 proteins that add up to like 3,000 residues, amino acids, and then you try to like dock them with AlphaFold, you might be running for like 1 day or 2 days. And so we have to be careful here. I would like to run this on like OS pool or something larger, but I have a problem where AlphaFold 3 has a licensing requirement that I've got to keep these weights that they provide you like hidden, and like I'm not allowed to like just send them anywhere. So I have to like figure out how to work around that. And I'm sure CHTC will help me with that. And so then I get like, hopefully I'll get down to 1,000 prediction interactions. I'll like make subnetworks and then try and build complexes. Hopefully we'll get around 50 complexes and maybe like 10 or 20 will be actually correct. And that will be a success, I think, for this project. I would like, just before we go to any questions, I'd like to thank Jacob Ekman, who came up with this problem, and Kevin Myers and Mike Place, who I work with, helped me with like feedbacks on the talk, and CHC facilitation team is great. They answered all my questions. Without which I'd still be stuck. Christina and Andrew. And the AlphaFold 3 pipeline is on here already. And thanks to Patricia for helping me with like some documentation and additional scripts. And so if anyone wants to use it, I wrote HPC instead of HTC. Yeah, that's a mistake on my part. I'm the wrong conference. And yeah, thanks to all of you for listening.
Thank you, Samir. I'm sure it was just a typo in your GitHub name. Christina?
Yeah. These are two very specific questions based on your last slide, Samir. So running on the OS pool, the step that you mentioned, does that need the 600 whatever database, or is that just for the data preparation?
No, that's just inference. It's only going to have, for pipelines, it's only going to have these alignments that I create,
Right.
which will be like in the megabyte range.
Oh,
and there'll be like efficient matrices, binary matrices that I'll just send across.
And then,
So
it's
a 3 gigabyte container.
Yep.
And that's it. And then GPU time.
Okay.
Yeah.
Well, we can talk.
And licensing for the weights.
Yeah. So you mentioned the coevolution step that you're basically reducing because you can't dock that many proteins. If you had 1 million GPUs, would you want to dock that many proteins?
Yeah. So what would happen is that if you just look over here,
or pairs,
instead of 50 complexes, I might get 80, and of which maybe 40, 30 or 40 are correct. So there is some benefit. But then there's a trade-off at how much that's going to cost, right?
Okay.
I mean,
yeah, I was like, if we could give you all the GPUs so you could actually dock all 1 million, would that be?
That would be useful, but we have to balance GLBRC's mission, which is about fuel economy.
Right.
Burning GPU. No, but yes, no, it would be useful.
Okay.
Yeah. If I could do this all with AlphaFold, then I wouldn't do any direct coupling. I just like do it all.
Okay, that's good to know.
So help me understand. You're running the docking. AlphaFold enables you to generate the docking information.
Yes.
And then you use the docking. You use the results to select the one that you run through the inference.
No, so AlphaFold just tells you roughly whether these proteins interact, because you only get a part of the docking interface. And then after that, we'll make a graph of all the proteins that interact, and then like find subnetworks, and then try and brute force those subnetworks into complexes. Because we have to figure out how many copies of the protein are in each complex.
So where's the inference coming in?
This is the inference.
This is the inference. That's what you call the inference. So the docking simulation is what you call the inference.
Yes.
Okay.
Yeah, if we try to actually run docking with traditional docking software, like each docking thing would take like maybe a day.
So you could do the docking with CPUs, but it would have taken longer?
It would have taken a lot longer, yes.
How much longer?
I don't know, because I basically work on machine learning stuff. So the other people who work on the biophysics-based docking. And I think you need to do a lot, because I think I did a little bit with Rosetta, not on this problem, but on a different problem. And basically, you've got to run, each simulation would take like 20 minutes, and you'd have to run like I'd say like 7 or 8,000 simulations to get one like good structure.
Yeah, because it's always an interesting trade-off that will help to understand is we can definitely bring many more CPUs to the table than GPUs.
Right.
So
Right.
at the end of the day, it doesn't matter how much each inference takes, you still have to do 20,000 of them. And
No, we have to do 1.7 million of them, but because of this step, we kind of cut it down to 20,000. This is a less accurate method of.
So you would have liked to do 1.8 million.
We'd like to do all of them, yeah. I mean, if
I understand.
free, we'd do all of them. Yeah.
So it may be an interesting question of can you go the CPU and do more but wait longer rather than go the GPU, or a mix between them, because we can definitely bring order of magnitudes more CPUs to.
But the CPU is order of magnitude faster than so the GPU is order of magnitude faster than the CPU.
Yeah.
It's about 100 times, 60 to 100 times, and things I.
So it's almost 2 orders of magnitude.
Yeah. Yes.
Okay, so but if rather than giving you 10 GPUs, we give you 10,000 CPUs, you may be better off.
If we can coordinate all of them together, maybe, yeah. But then
Yes.
then there's also like communication between the CPUs. Yeah.
They need to talk to each other?
Well, if you're going to split one in, like these are large matrices, right, that get multiplied over. So if you're going to split them across CPUs, then they have to talk to each other.
I said the CPUs. Okay, I would like to better understand. Okay. Let's keep going.
Okay, let's thank Samir again.